Okay, perfect. So today we have the pleasure of having two speakers for our PacVec seminar series, and this is a first in our series to start integrating our training grantees, which is a very exciting element of our program, as you all know. And today we have Dr. Tara Thiemann from the University of the Pacific, who will present during the first half of the hour, and then she'll be followed by Kacie Ring from San Francisco State University for the second half of the hour. I think the way we will work this is to reserve questions for each speaker until the end of their talk. And you can ask questions by either typing them in the chat window or by asking at the end of the presentation. So just hold your questions until then. And with any luck, we'll be able to allow the speakers to share their screen and present for about 20 to 25 minutes, and then we'll take the break for questions. So first up, we have Dr. Tara Thiemann. She's an associate professor of biological sciences at the University of the Pacific. She got her bachelor's and master's degree from Truman State University in Missouri, then took a brief break, I believe, in between those in Australia for a bit, and then pursued her PhD at UC Davis, where she worked with Bill Risen and studied blood feeding patterns in West Nile virus vectors. She also developed a number of molecular tools that we're still benefiting from here in the lab at UC Davis. And then in 2012, she moved to the University of the Pacific, where she took a job as a professor. She's already mentored 10 grad students there, and 2 of those grad students so far have been PacVec training grantees, which are Sumiko de la Vega and Bonnie Ryan. Their projects have been an excellent example of our community of practice that we aim for in PacVec, where Tara works closely with mosquito control agencies. Those agencies also contribute to her work, and the students get experience and even sometimes careers in some of the vector control agencies in our state. So with that, Tara, I'll turn it over to you to talk about pyrethroid resistance and Culex tarsalis.
Great, thank you. Let me see if I can get a screen shared here. Okay. All right, so I think then if I keep the window here, will that go to me? Are most people seeing me in the corner?
It looks perfect on my side. I can see you in a window to the side of this larger slide.
Perfect. All right, so first of all, thank you for having me. I am presenting on behalf of Sumiko and Bonnie. So they were the PacVec trainees from a 2018 grant. They started this work on resistance and tarsalis then, and they're still working on getting sort of data analyzed and feverishly writing masters right now. So I agreed to talk today on their behalf. I also want to thank those vector control districts that Chris mentioned. So this was a close collaboration with San Joaquin, Saciolo, Placer, and Lake County. The project is absolutely not possible without them. They picked study sites, collected mosquitoes, have interacted with us about data analysis, and also provided funding. The primary funding, of course, came from PacVec, and then I also have some funding from the University of the Pacific. So why study Culex tarsalis? It's a native mosquito to California, so probably many of us on this call have been bitten by it more than once. It is an important vector of West Nile virus and other arboviruses. Historically, it's been documented as a rural mosquito, but more recently, it's also been found in urban areas. This is in part because of residential areas moving into rural space, but also the mosquito actually moving into residential areas with things like abandoned swimming pools, etc. So because it has both this urban and residential footprint, it faces a lot of insecticide pressure from agricultural spraying, as well as from vector control. And until recently, I'd say until about 5 years ago, few reports of insecticide resistance had been documented for this species. All right, so project goals. In working with the vector control districts, they were starting to see some resistance, or at least what they thought was some resistance developing in this mosquito. So the first thing we wanted to do was determine the prevalence of resistance, particularly to pyrethroid. So this study, we're looking at pyrethroid resistance in tarsalis. We expected we would find some resistance, and so then the remaining goals of the project were to characterize that. So was this resistance due to enzymatic resistance, target site mutations, or perhaps both? A quick review on how pyrethroid insecticides work. So first of all, they target the nervous system of the mosquito. They bind to and open voltage-gated sodium channels. This increases an influx of sodium ions into the cell. This then increases the number of action potentials and then the amount of acetylcholine, the neurotransmitter that's released at the synapse. With this, we get increased neural transmission as well as muscle contraction, and basically the mosquitoes use up a lot of their energy for these processes, and they undergo what's called knockdown, and eventually they die. Now, there are a couple of different mechanisms that are primary insecticide resistance mechanisms. The first one, detoxifying enzymes. This chart diagram here is modified from a Brogdon-McAllister paper, basically looking at different classes of pesticides and what enzymes have been shown to lead to resistance. We're focused on the pyrethroid, so you can see that esterases, oxidases, and GSTs, there's been evidence that they may assist or lead to resistance in mosquitoes. The other primary mechanism is target site mutations. These are DNA mutations that lead to amino acid substitutions. In this case, for pyrethroids, our mutation of interest is something called KDR, or knockdown resistance. This is a mutation in the voltage-gated sodium channel. Basically, the mutation prevents the pesticide from binding or from binding as well, and it can't lead to its effects. Now, there have been recent studies that show that probably there are several mutations that can lead to resistance. The primary one and the first one discovered is this one that's found at codon 1014. And there have been two different mutations at this site documented, a leucine to phenylalanine and a leucine to serine. This is the only mutation that we looked at for this particular project. All right, so back to our goals. First, we want to determine prevalence of pyrethroid resistance. We did this using CDC-style bottle bioassays. We collected mixed age female tarsalis from CO2 traps, and our goal was to hit 3 to 5 populations per district so we could get a nice spread across this region in Northern California. For each round of bottle bioassays, we tested permethrin, which is a common pyrethroid insecticide. We also tested permethrin plus PBO. Now, PBO is a synergist. It inhibits oxidases. So this is often combined with pyrethroids when the pesticide is sprayed to help sort of limit any resistance that may come from the action of the oxidases in the mosquitoes. We also had our control bottles, PBO only and acetone only. Acetone is the chemical that you use to deliver the pesticide to the bottle. So basically, this acetone only is an empty bottle, the acetone's evaporated, and there's no chemical residue left. Every time we did bottle bioassays, we tested a susceptible colony along with our wild-caught populations. We had 2 bioreps per population, and we put about 20 mosquitoes into each bottle. After the mosquitoes were in the bottle, we recorded mortality every 15 minutes or so. We went to 120 minutes, and at that point, we separated the dead and the living mosquitoes and marked those for later testing. Now, the data from the bottle bioassays we can show in these mortality curves. On the mortality curve, you can see percent mortality on the y-axis, you can see time on the x-axis, and for each of these, there are going to be 4 lines. We have the colony with and without PBO and the wild population with and without PBO. Now we have 17 populations, and I'm not going to show them all to you today, but I want to show you a few examples that demonstrate kind of the variation in resistance that we saw. So first, this Empire site from San Joaquin County. Green is the susceptible colony. You can see that the colony died in 15 to 30 minutes as expected. The wild type population in this area, eventually the mortality did near 100%, but you can see it took much longer than it did for the colony. When we add PBO, mortality is reached at 100%, back closer to the colony at 45 minutes. Another example from a site in Placer County. This time we used the KNWR colony. Now, in an ideal world, we would have used the same colony for all of our testing that summer. Tarsalis is notoriously difficult to keep in colony. And that summer, it turned out that a couple of colonies around the state had collapsed. So we were really scrambling kind of on a weekly basis, even to get susceptible colony that we can use. So that's why we used a variation in the colony. One thing interesting to point out is that at the same dosages of pesticides, you can see it took a little longer for KNWR to die. Still though, it died within 45 to 60 minutes. Looking now at the wild population, only about half of the mosquitoes died in this round. We had PBO though, and mortality again nears 100%. Finally, one of our more resistant populations, this is a population from Yolo County. We can see that the KNWR is behaving about the same. Mortality for permethrin only was only 8.6%, showing quite a bit of resistance in this area. And even when we added PBO for this one, while mortality certainly increased, it did not get above 90%. So here's a table summarizing those 17 study sites. You can see that there weren't as many study sites in Lake County. That was a summer that there was a big fire in Lake County. We were only able to go up and do bottle bioassays once. We had planned a couple of more trips. So we added some populations to San Joaquin and Sac-Yolo to make up for that. I'm hoping to get up to Lake County this summer to do a little more testing, but we'll have to see how the rest of the summer shakes out. All right, focused on mortality in permethrin only bottles. So these are percent mortality after the 120 minutes. So the higher the mortality, the more susceptible the population. You can see we had some populations that had pretty high mortality, 96.2% in this case. We also had populations with really low mortality with permethrin only. This one from Vine in San Joaquin, only 1.5% mortality. And if you scan through the rest of the numbers, you can see there's quite a variation in what percentage of the mosquitoes died during the bottle bioassay. Now there's some good news when we look at the permethrin plus PBO. So again, this is the synergist that inhibits oxidases. Most mortality percentages increased quite a bit here. In fact, most of them went above 90%, though we still have that one population that didn't quite get to the 90% mortality. Now, this data with permethrin plus PBO gives us a clue that oxidases are likely playing a role in some of this resistance, because when you add the oxidase inhibitor, mortality increases. So wanting to further investigate those oxidases and also look at other detoxifying enzymes, we move on to assess enzymatic resistance. Quickly on the methodology for this, you basically take the mosquito and you grind it up with beads. You get this mosquito homogenate that's going to have the enzymes in it. You put those into a 96-well plate and add a substrate and other reactants. Basically, the assays were designed not by us. This is previous work. The assays were designed so that the enzymes take a substrate, convert it into a product that has a color, and then that color can be read in a plate reader, which is essentially a spectrophotometer. So you can see on the right in this diagram, you would expect to see color change depending on the activity of the enzyme. Okay, so using standard CDC protocols, and these are protocols that are used by the Sac-Yolo district as well, we tested for oxidases, alpha esterases, beta esterases, GSTs, the levels of acetylcholinesterase. We also looked for insensitive acetylcholinesterase, so that is more an organophosphate mechanism. And then finally, we did measurements of total protein. This helps us normalize all of this data for mosquito size. All right, so I'm going to show you just a few of these to kind of give you an idea of where we are. As I said, both Sumiko and Bonnie are still working on data analysis, so I'm just going to give you a kind of a quick snapshot. So here we are looking at data from San Joaquin. These are alpha esterase levels. So on the y-axis, we have the alpha esterase activity normalized by protein. So again, that helps us normalize by the mosquito size, and then across the bottom, you can see the population. The first box plot there is BFS, and that is our susceptible colony. So probably what will strike you first, or what certainly struck me first, is that BFS actually has higher alpha esterase activities than the rest of the wild populations. We absolutely did not expect this. It's a susceptible colony after all. So if we ignore that for a moment and look at the other populations, we can see that there is an increase in alpha esterase for Empire and Vine. Those letters across the top are denote statistical significance. Moving on to beta esterases, we see a similar pattern. So first of all, that susceptible colony was super high levels compared to everything else. And in this case, there's not a huge amount of difference between the wild caught populations. This issue with the BFS colony, and I'll just say, though I don't have the data to show you that we see it in the KNWR colony as well, this provides some issues for us because the way that this data is usually presented is something like the proportion of a population that has higher levels than the susceptible colony. That's how the data is presented to see if there is overexpression of these enzymes. And obviously we can't do that if we have a susceptible colony that has higher levels than the rest of the wild population. So we're still kind of working through how to deal with that. A couple of more enzymes that I want to show you though, oxidases. Now this one we expected to have higher oxidase levels because we know that when we added PBO to the bottle bioassays, we got higher mortality. So that would tell us that oxidases are at play here. You see then that there are, again, a couple of populations, that Vine and Empire population, that do have some outliers and do have some increased expression, but they look pretty even with the susceptible colony. Now, I don't know this for 100%, but what I think at this moment, and we're working on trying to figure this out, is that BFS does actually probably have elevated levels of many of these enzymes. And what we're seeing here is not that BFS is lower so that it matches the wild populations, but in fact, everything we're looking on this graph may have higher oxidase levels compared to a susceptible wild population. Again, this is causing a bit of a struggle for us, and we're still working on how to figure out how to interpret some of this enzyme data. Moving to the last one that I wanted to point out, this is the levels of acetylcholinesterase. We can see that there are several outliers, right? Individuals that are expressing a lot of acetylcholinesterase in these populations. And again, while not 100%, I'm thinking that these wild populations probably do have higher levels of acetylcholinesterase. It just happens that the BFS colony does as well. Okay, so two things to kind of summarize this. First of all, we're not at all happy with the colonies and being able to use the colonies as an enzyme reference. So this summer, assuming that we can get there with everything going on with COVID-19, a plan is to look for a more susceptible wild type population so we can further assess these enzyme levels. We did want to see what we could do without using BFS as a reference. And so talking to John Mayberry, who's a statistician at Pacificus campus, we decided to try some regressions. So basically here we have survivorship on the y-axis, and I use survivorship because then that's a positive correlation with resistance. And we have the 90th percent of enzyme levels on the x-axis so that we're catching those high levels of enzyme from individuals in the population. These p-values denote whether or not the slope of the lines are significantly positive, right? So is there a positive correlation? This is very, very preliminary, and we're hoping to add more populations to this in the coming months. But looking at oxidase, we can see it borders on significant, and we have some reason to think that oxidases are playing a role here. It also is a potential then that GSTs and acetylcholinesterase are positively correlated with survivorship in our bottle bioassays. All right, moving on to target site mutations. So I mentioned for pyrethroid resistance, the target site we're interested in is KDR, and this is a mutation in the voltage-gated sodium channel. We used a process where an assay where you can use cyber green with these GC-rich tails on the primers. So the primers are diagnostic for the SNPs for the KDR location, and then we've added these GC-rich tails so that it artificially modifies their melt temperature. When you run the assay and the melt curve, you get data that looks a little something like this. So phenylalanine alleles have the lowest melt temperature, leucine in the middle, and serine on the right. Now you can probably see that that serine has a pretty clear 2 bump. That's because despite our best efforts at this point, we still have the leucine primer binding somewhat to a serine template. It's not that huge of a deal. We can't distinguish LS from SS individuals, but we do have a secondary test so we can sort that out. And there's not a huge number of individuals that fall into that category at this point. All right, so what does that data look like? Again, just giving you a snapshot, I'm going to show you what Sumiko has put together for San Joaquin. In all of these pie charts, we're looking at allele prevalence. So the green is the leucine allele or the wild type allele. Phenylalanine and serine, the F and S here, those are both known KDR alleles. Let's start with the BFS colony. First of all, very good news. That is all of the alleles came back as the leucine wild type, which we would expect for the colony, but given the enzyme data, we were very happy to see this. Now, if you look at the rest of the wild populations, you can see the story is not quite as great, right? So definitely the most prevalent allele in this area is phenylalanine, which tends to be the most common KDR allele when you look at other mosquito species and other papers as well. And we also have a number of the serine allele in these populations too. Now, the data from the rest of the state I don't have put together as well right now to show you, or from the rest of northern, the areas I should say, I don't have put together as well to show you, but the trend is pretty much the same. These KDR alleles are fairly common. So then I started to think, well, we were pretty sure there was going to be some resistance, but to be honest, we didn't expect this much resistance. So I wanted to ask, I wonder how long these alleles have been circulating in these populations. Well, it turns out that I have Culex tarsalis that were collected while I was a grad student, so 10 years ago, give or take, from very similar areas to the ones we're testing now. I'm teaching a vector-borne disease class this spring, and we did a research project. So this is a little bit of data from that vector-borne disease class. Let me go ahead and pop up the graphs here. Okay, so I picked nearby sites that I had samples that were collected in 2006 to 2008. That's while I was a grad student. And then I have more recent collections from my current master students and previous master students that were collected in 2006, from 2016 to 2018. I picked sites that were close together. We have one matched pair of sites in Yolo County and one matched pair of sites in, from the Sutter Yuba district. And you can see there's been a pretty big shift in these KDR allele frequencies or these KDR allele prevalence over that time. So in 2006, 2008, that leucine or wild type allele was very much more prevalent than it is today. And you can see that there's been this big shift to the phenylalanine allele being most prevalent in these areas. I was, however, interested to see that even in 2006, 2008, there was a fair proportion of those phenylalanine alleles already circulating in my Sutter Yuba study sites. Okay, so to summarize, there's substantial resistance to pyrethroids in tarsalis in our 5 study counties. Mortality ranged from 1.5 to 96.2% in our bottle bioassays, but for the most part, adding that oxidase inhibitor brought mortality above 90. The enzyme data is a little cloudy because we're not sure how to do comparisons with our current susceptible colony. However, there's a potential that there's increased oxidases, GSTs, and acetylcholinesterase that may be affecting resistance. And there's a high prevalence of leucine to phenylalanine mutations at that KDR allele and some leucine to serine mutations as well. And I think it's pretty clear that the prevalence of these mutations has increased greatly in the last 10 years, though I hope to increase my sample size and continue to look at that further. And with that, I am finished, and I'm happy to take any questions.
All right, thank you, Tara. That was great.
And I think we do have a question from Jay Gann. So Jay, I will unmute you.
Okay, thank you for the nice talk. It's I have a question just based on your research, also based on your knowledge. If we have to pick up one enzyme to do an assay for pyrethroid resistance, what would that enzyme be?
Can I pick two? I think that we have the most evidence so far that for pyrethroid resistance, oxidase is playing a role, but that's in part because we have multiple ways to look for it, right? So we have the bottle bioassays where we can check to see if PBO makes a difference, and then we have the enzyme assays. The one that I'm actually most interested in at the moment are the levels of acetylcholinesterase, and that's in part because it's not something that's normally looked at. So that's the one that I'm focused on now to see if there's really something there or not, because it could be a mechanism that we haven't thought of before.
Okay, thank you.
Mm-hmm.
All right, I see a question in the chat from Sarjeet Gill who's saying, well, let me remove my other window. What was the substrate used for the oxidase assay?
So apologies, but I don't remember right off the top of my head. I can definitely look it up and email to you. We're using the standard CDC protocol that was then put together by the MVCAC working group. I can't remember the exact title of it, but it was a previously published assay, and I can certainly look up the substrate for you.
Tara, one other question I had about the distributions within populations that you see for the enzymes. Is there anything you can make out of the right tails to some of those distributions where some of them have very high values that are clear outliers compared to the general center?
Yeah, so from a statistical standpoint, I've been struggling with how to deal with those, because in most statistical analysis, you want to bring those in, right? But really, I think those are probably the interesting individuals. So I think what we may be seeing when we have, some mosquitoes, like a small proportion of mosquitoes that are surviving insecticides, it may not be a general shift in the whole population, but rather some proportion of individuals that are high expressors.
Yeah, very interesting. All right, are there other questions? Looks like we have one more in the chat. Have you considered that there might be a mutant aliesterase involved in resistance that could account for the lower esterase activity in your resistant strains compared with your susceptible strain?
One more time, the first part of that. Sorry, I can't see it.
Have you considered that there might be a mutant aliesterase involved in resistance that could account for the lower esterase activity in your resistant strains compared with your susceptible?
So an esterase that the standard protocols is not measuring the activity. I hadn't thought of that, but that's certainly something to keep in mind. Yeah, and that could be why the colony is so high and everything else is so low. Yeah, certainly something to think about.
All right, one more. It says, for the insecticide resistant assay, do you correct for the age difference between lab and wild collected mosquitoes?
So that's a very good point. So in other mosquito species, you probably know the standard is to use 3 to 5 day old adults. Because it is so hard to rear tarsalis, that hasn't been the standard. So what we did is we did mixed aged adults, but we did our colony as mixed aged also. In addition to that, while I didn't show the data today, Samiko did a project where she compared mixed age to various age groups. And this is super off the cuff, right? So I'll have more to say about this later, but in that limited study, it looked like the biggest differences were if you had very young mosquitoes. So if you had mosquitoes that were kind of 0 to 2 day old, they were significantly different from the mixed age, but the other ones weren't. But the short answer is, for the colony, we used mixed age so that they matched the wild populations.
All right, so in the interest of time, I think we'll move on. Thank you very much, Tara. That was a great presentation. And I think if anyone else has questions for Tara, if you are able to see the chat, Tara, if you want to answer those
Yeah, I'll
as we
see if I
go.
can get to it after I close out of here. Thank you.
Okay, thank you. And Frank, who asked the question about the oliesterase, also makes the point that that does occur in dipterans, so maybe worth further investigation. All right, so next up we have Kacie Ring. Kacie is at San Francisco State University, as I mentioned before. She is currently a recipient of one of our PacVec training grants for the current year for 2019-2020. She graduated from UC Santa Cruz with a bachelor's in ecology and evolutionary biology. She is currently working in Andrea Sway's lab at San Francisco State, where she's pursuing a master's degree in microbiology. She has a wide range of field and lab experience, from small mammal trapping to lizard noosing to next generation sequencing and other molecular work in the lab. So with that, I will turn it over to you, Kacie, where she will be talking about host blood meal's influence on the vectorial capacity of Ixodes pacificus.
Yes, I'll share my screen. Okay. All right. So part of my research, so part of my PacVec training was involved determining the LC50 of permethrin and Ixodes species. And that was in collaboration with San Mateo Vector Control. That research is more applicable to vector control research and the tenets of PacVec, but PacVec has also funded a more exploratory side of my thesis, of which I've recently gotten some pretty interesting results. So I thought that that would be more interesting to present to everyone today. So the title of my thesis is Host Blood Meal's Influence on the Vectoral Capacity of Ixodes pacificus. And so vector-borne diseases around the world are emerging. I'm sure I don't have to tell this group that. But interestingly, if you break down the vector-borne diseases by vector, ticks actually harbor the most diseases out of all vectors. And the map on the right here shows the distribution of key tick-borne diseases in the United States. Now you can see that most of the tick-borne diseases are concentrated on the East Coast. And you can see that the color of the map that is most prominent is purple, and purple represents Lyme disease in this case. But if you look over in California, you can see a bit of a concentration of purple in the Bay Area, which is where our lab does our research. And so a little bit more about Lyme disease. It's vectored by the black-legged tick, Ixodes species. On the East Coast, it's Ixodes scapularis, and on the West Coast, it's Ixodes pacificus. And it's a zoonotic disease maintained by ticks and a wide variety of vertebrate hosts like mice and rats and squirrels and birds. And the pathogen that causes Lyme disease is a bacterial spirochete, Borrelia burgdorferi, which I may refer to as BB throughout the rest of my presentation. And so one of the advantages of studying ticks is their seemingly straightforward life cycle. So they have 3 life stages, larvae, nymph, and adult. And they live for about 2.5 years, and and the larvae and the nymphs feed during springtime, and then the adults emerge sort of late winter. And since Borrelia burgdorferi cannot be vertically transmitted from adult to progeny, or transoverly transmitted, larvae emerge uninfected. So these blood meals that the ticks have at each life stage are discrete opportunities for pathogen transmission. And what complicates tick-borne research is that the hosts that they feed on are very different. They're very generalist species, so they can feed on a reptile, they can feed on a small mammal, they can feed on a larger predator. So the host blood meals that they intake are all very variable. And so this graph is a little bit busy, but I think it's really interesting because it looks at the role of hosts in all these areas that are common to have Lyme disease. And so I want to focus on the West Coast, where Ixodes pacificus is. And this bar graph basically shows, in the first bar, it shows the proportion of ticks fed by that particular host. The second graph bar shows the relative reservoir competence of the host. And then the third bar graph shows the proportion of ticks that are infected from that host. And so each color represents a different host. And you can see that there's this really large gray bar in the first column for the Ixodes pacificus, and that represents the western fence lizard. And you can see that the gray bar does not carry over to the relative reservoir competence or the number of ticks infected. And that's because the western fence lizard can't infect ticks and has no relative reservoir competency for Borrelia burgdorferi. And we also see that translated into our field data. So on the left is a graph of the mean tick burden on our common hosts. And you can see that lizards have a mean tick burden of about 25 ticks, but we've collected lizards at our field site that have had over 100 ticks on an individual lizard. So those numbers are really variable. But it's not as variable with wood rats and mice. We generally see, 0 to 8 ticks on each individual, wood rat or Paramyscus mouse species. And so I'm also going to probably talk about these hosts in there. We have a lot of acronyms for things. And so we commonly refer to the western fence lizard or Scoloporus occidentalis as SCOC, just the first 2 letters of the genus and species. Neotoma fusipes, or the wood rat, is NIFU, and Paramyscus maniculatus is PIMA. So if you hear those acronyms, they just correlate with the animal. And so, as I've mentioned before, hosts vary greatly in their reservoir competency. So squirrels and wood rats are very good at transmitting Borrelia to ticks. Mice are okay, and lizards cannot transmit the disease. And they also have a borreliocidal effect. So if an infected tick does feed on a lizard, it will end up being cleansed of its Borrelia burgdorferi infection. So not only they can't transmit it, but they also reduce it. And so that kind of sparked an interest for me in looking at the role of lizards in Lyme disease ecology. While they're a super important tick host, supporting huge populations of, juvenile ticks, they are also an extreme dilution host. And our lab was also interested in knowing if this borreliocidal effect that the lizard blood has on the tick is also a bactericidal effect. And before I got there, Andrea already did this study looking at the microbiome composition between ticks that fed on Paramyscus mice and ticks that fed on lizards in their larval blood meal. And so when they molted into nymphs, she did a 16S microbiome composition study and found that lizard-fed ticks had a significantly lower richness, as well as significantly different bacterial communities. And you can see that's kind of reflected in the composition of the OTUs. Both are dominated by the Rickettsia endosymbiont, but you can see that the richness is greatly reduced in the SCOC or lizard-fed ticks. And so now there is this natural variation in microbiome composition in natural hosts. Other studies have looked at how the microbiome composition will actually affect future pathogen acquisition. So this study was done out of Yale, and it looked at the effect of a disembodied or reduced microbiome on future pathogen acquisition. So they experimentally reduced the microbiome by feeding larval ticks on gentamicin-treated mice, and they found that the disbiased ticks actually ended up having lower pathogen acquisition in future blood meals. This is getting a little complicated, but it'll make more sense when I explain my research as well. And so they conceptualized that the reduced microbiome ended up downregulating certain humoral genes, innate immunity genes like STAT, which is a transcriptional regulator of the JAK-STAT pathway, which in turn decreased peritrophin. And the peritrophin membrane in ticks has shown to be very important for Borrelia colonization. Basically, if you see over here in this little purple image, it's a very thin epithelial that separates the gut lumen, which tends to be very acidic because ticks only feed on blood, and the epithelial cells. And so just to kind of simplify this conceptual model, it's basically higher microbiome composition leads to a thicker peritrophin matrix, which acts as a refuge for Borrelia burgdorferi. And so when we have lower microbiome composition, this peritrophin matrix thins, and then the Borrelia does not have enough room to colonize and dies with the acidic pH from the gut lumen. And so if I were to directly take the results from the study at Yale and hypothesize what will happen with ticks that have these different larval blood meals, I would assume that the lower microbiome diversity associated with this lizard blood meal would then lower the colonization of Borrelia burgdorferi in my ticks. And this lizard blood meal will then reduce pathogen acquisition in future stages. So I have two goals of my thesis. The first one is, how does host blood meal affect future pathogen acquisition? Does the lizard blood meal reduce future pathogen acquisition if they feed on an infected host, or does it increase it? And I would guess that due to the reduced microbiome diversity, we should see reduced pathogen acquisition compared to ticks that fed on mice after they feed on an infected host. And then goal 2 would be, if that was true, if host blood meal legacy does affect Borrelia burgdorferi transmission rate, what are the underlying mechanisms? And I would say that the underlying mechanisms would probably be associated with downregulation in genes associated with immunity and gut morphology after their nymphal blood meal on a Borrelia burgdorferi infected host. And so my experimental overview is that I collected larvae off of lizards and mice and allowed them to molt into nymphs in lab. I then fed the flat nymphs on C3H mice. Half the mice were infected with Borrelia burgdorferi and half the mice were uninfected with Borrelia burgdorferi. And then after the nymphs completely fed, I waited 24 hours, flash froze them, and then extracted the DNA and RNA from the whole nymphs. And so goal 1, I'll use the DNA to determine presence and load of Borrelia burgdorferi via qPCR. And then goal 2, I wanted to create RNA-seq libraries and compare 6 experimental groups, which I will describe in a little bit. And so for my larval feeding, I went out and noosed lizards at Hastings and Tiburon, a couple field sites that we have. And then I only took lizards with really high burdens and brought them into lab spaces to do natural drop-off experiments. Previously, we would pull ticks off with tweezers, but we found reduced molting rates and reduced mortality or higher mortality. So doing the drop-off experiments allows for natural engorgement. And then I kept the engorged larval ticks alive in desiccators. This was kind of my MacGyvered lab setup at one of my field sites. I can't take the lizards too far, so I have to kind of make do with the lab space that I have. But I had 5 lizards with and some of them had over 100 ticks on them. So it was easy to get high sample numbers. In comparison to mice, they have super low larval tick burdens, so it would be impossible essentially to collect the Peromyscus from the field and do these natural drop-off experiments. So I ordered mice from the Genetic Stock Center and then experimentally fed larval ticks at the animal facilities at SFSU. And then I collected the ticks in the same way. After they molted into nymphs, I inoculated my C3H mice with the California strain of Borrelia burgdorferi. The nymphal ticks feed better on the C3H mice, and the C3H mice don't groom as much as other mice species, so that's why I chose them to be the nymphal blood meal. And then I did the same drop-off experiment with the C3H mice, and nymphal ticks from both blood meals, lizard and mouse, were fed on the infected and uninfected C3H mice. And then I did drop-off experiments. And so I froze them after 24 hours and then extracted their DNA and RNA, and the DNA was used to determine the pathogen load in the ticks that fed on the infected mice. And so basically, I found the exact opposite to be true than what I hypothesized. So ticks that I fed on lizards as larvae and then on an infected mouse as a nymph had a way higher infection rate than ticks that fed on paramyscus mice and then fed on an infected C3H mouse. Basically, 93% of the ticks that fed on the lizard as larvae were infected, whereas 23% of the ticks that fed on paramyscus as larvae were infected. And we ran a GLM with experimental trial as random effect and found that there was significantly high pathogen transmission in our lizard-fed ticks. I also want to get the Borrelia load with qPCR, but unfortunately, our standards were created after I did the initial test, and now I can't get back into lab to look at the loads, but hopefully I'll be able to get back in to figure out the general load of the pathogen. But it looks like from the CT score that there's way higher loads in the lizard-fed ticks as well. And so my initial hypothesis was wrong. It was the complete opposite. So now I kind of had to move on to another hypothesis. So maybe the lizard blood meal increases vectoral capacity of Ixodes pacificus through suppressing tick immunity and increasing pathogen transmission in the nymphal blood meal. And so now I'm able to maybe figure out what the mechanism is behind that differential pathogen acquisition by doing an RNA-seq study and looking at that transcriptomic profiles. So I have 6 experimental groups, all kind of controlling for different things. So the first 2 groups are unfed ticks that had this, larval blood meal of a paramyscus and a lizard. Basically, these acronyms, the first 2 letters indicate the nymphal blood meal. So unfed, mouse-fed, Borrelia, just so you can kind of wrap your mind around these weird acronyms that I have. And then the 3rd and 4th groups are the mouse-fed uninfected ticks that have paramyscus and larval or lizard larval blood meals. And then the 5th and 6th group in my transcriptic profiles are the Borrelia burgdorferi infected mice that fed on, or ticks that fed on mice and lizards. And so I have all these groups. The uninfected mice control for genes associated with feeding, and then these control for genes associated with infection, and these control for genes associated with feeding. So I need to have all these groups to just control for differential gene regulation. And so for my transcriptomic analysis, I checked the quality and quantity of my RNA with a bioanalyzer before prepping my libraries. I prepped my libraries with the NEB-Next ultra-directional RNA kit, and I had to prep with total RNA, and then I used a DASH protocol made by collaborators at UCSF that targets the rRNA with CRISPR for tick, mouse, and Borrelia. So then I ended up with just mRNA in my samples. And then I did the high output sequencing on an Illumina NextSeq at BioHub. And then I mapped my sequences to the newly annotated ISE6 Ixodes scapularis transcriptome using a software called Salmon. And then I used DSeq2 for differential gene regulation. And so the 2 transcriptomic profiles that I'm mostly interested in are the nymphal ticks that fed on the infected C3H mice, because I want to know what the mechanism is that's causing the differential pathogen acquisition in my ticks. So could the paramyscus ticks be mounting a stronger immune response, or are the lizard-fed ticks more vulnerable somehow? Just wanting to figure out what that mechanism is. And so this is a volcano plot. And all these weird numbers are the gene ID associated with what it mapped to. And so basically, whatever is upregulated or downregulated are the associated with the lizard-fed ticks, and the mouse-fed ticks are the control in this case. And unfortunately, this doesn't show that many genes being differentially regulated. And so when I kind of break it down, most of them are uncharacterized. That's one of the problems with working with something that is not a model species, is a lot of the genes, almost majority of them are uncharacterized. And none of the genes that are differentially regulated show any sort of immunity signals. So I was a little bit bummed about this result at first. And so I'm realizing maybe it's not, there's no sort of suppression of tick immunity. maybe there's something going on in the larval blood meal that could have an impact on future pathogen acquisition. So I wanted to look on what was going on in my unfed nymphs also. And so this volcano plot shows a ton of differentially regulated genes. Now this group is just the ticks that fed on lizards as larvae and just the ticks that fed on mice as larvae. So they didn't have a nymphal blood meal. This is just the differences between the larval blood meal groups. And there's a huge difference, way different than the nymphal blood feeding groups. And when we look at what genes are actually being upregulated and downregulated, a lot of them are showing immunity signals. So just to remind you, everything that is upregulated or downregulated are the ticks that fed on the lizards. And so we see upregulation in antimicrobial peptides, defense genes, genes associated with blood digestion, and genes associated with morphological changes. And something that's really interesting to me is that the most upregulated genes in the lizard ticks are these antimicrobial peptides, acanthoscurin 1 and acanthoscurin 2. And these AMPs have never been described in ticks before. The only sort of indication that I can find of them in the literature is from tarantula spiders. So that's the only time they've been described. And we also sent some of our unfed ticks off for histology, looking at cross sections of the tick gut. And in our lizard fed ticks, these are just no nymphal blood meal, just the larval ticks. You can see these really interesting brown granules that are forming in the midgut versus the mouse fed ticks don't have any indication of those brown granules. And this strange antimicrobial peptide that's being upregulated in my lizard fed ticks is produced in the hemocytes, so the blood cells of a tick, and then stored in granules. So perhaps this is all brand new data. Also, I've had my sequencing data for about 3 weeks, so I'm just kind of hypothesizing right now. But the antimicrobial peptide maybe isn't working against Borrelia. It could actually be reducing microbes in the tick midgut and helping Borrelia. That's just a hypothesis. There's nothing showing that. So just initially, since this is kind of new data, I'm guessing that the larval blood meal has a really significant effect on gene expression, more than the nymphal blood meal does. And despite significantly different rates in pathogen acquisition between groups with different host blood meal histories, few genes are differentially regulated. So that group that I showed you, both of them that fed on the Borrelia infected mice, maybe that's not what is causing this differential pathogen acquisition. And then could the larval blood meal be what's responsible for enhancing a nymph's vectoral capacity? So right now I'm just working on exploring those mechanisms. And even though I don't have a lot of time left in my master's, I have some future directions. Like I want to categorize all the differentially expressed genes using a gene ontology program to kind of figure out the function. And I also want to incubate Borrelia burgdorferi with that interesting antimicrobial peptide to see what their interactions are. And it obviously doesn't harm Borrelia, so I want to see if it kind of has a symbiotic relationship. And then I want to do more histology, taking partially fed ticks on lizards to see the effect on the peritrophin matrix. And then I would love to do a microbiome composition in all my experimental groups to see if that changes. So I'd really like to thank my funding, PacVex, so much, and my PI and Angie. I know you didn't have a role in this experiment, but you really helped me so much with my LC50 experiment and all my lab members. So if you guys have any questions, I will gladly take them now.
All right, thank you, Kacie. Are there questions?
I have a question. I don't know how to raise my hand.
We hear you.
I'll just ask it. So when you had, so the ticks that fed on mice were exclusively colony sourced.
Mm-hmm.
Do you have any ticks off of wild-caught Paramyscus to compare?
No, I don't. And so that was something that we realized is a big problem in this because, the pathogen acquisition differences could be, due to the wild versus colony ticks.
Yeah.
But doing some more like literature-based research, I found that that there's been studies showing that laboratory reared ticks have lower microbiome diversity generally than wild caught ticks, and that they tend to have higher pathogen acquisition rates. So if that had a huge effect on my samples, then it would be seen as the opposite being true. Like they would have more pathogen acquisition than the wild caught ticks. So I can't say that for sure, but we also want to do a study on looking at the transmission rates between wild caught and lab reared ticks. That's like another experiment we want to do.
Is another layer. Yeah, I think that'd be really cool. Just
Yeah.
because they're rabbit adapted over so many generations or whenever they last supplemented their colony, like how rabbit adapted they are versus mice or anything else is.
Yeah, for sure.
Thanks, that was super cool.
Thank you.
Kacie, I guess there's a lot to consider epidemiologically when you think about the potential effect of the blood meal on the tick itself, the microbiome, the Borrelia pathogens. What do you think then? I know your lab has done some work. Bob Lane's lab years ago did some work also showing the Borrelia-cidal effects of lizard blood.
Mm-hmm.
In terms of the net effect on the tick and the capacity for transmitting pathogens causing Lyme disease, what do you think that is, taking everything together?
So I think that the lizard blood meal has a sort of almost antibiotic effect on the tick gut. And the previous research indicated that that would actually reduce pathogen acquisition, but I think it's actually reducing microbe competition, essentially. I think it's kind of opening up a niche for the Borrelia to colonize. And I don't think that it's necessarily like having an effect on gut morphology or anything. I think it is just kind of expanding the opportunity for Borrelia to colonize. And that's kind of what is causing the differential pathogen acquisition. But I also don't know if that would have a big effect on Lyme disease prevalence in the field, because this is happening, they have to feed on a larvae and then feed on an infected nymph, and then it would emerge as an adult. So I don't think it would have, this lizard blood meal isn't going to have any implications for human health in the context of Lyme disease, because it would just be the adults. But I do think that the lizard blood meal does have a really interesting effect on the, tick vector competence that could be used for looking at novel control strategies and stuff.
What about survival and densities of ticks as a population? Like, what does feeding on a lizard versus feeding on a mouse mean to the tick?
So actually, we have found that lizard-fed ticks feed way longer. So when I would do the drop-off experiments, the pacificus ticks would take 3 days, whereas the lizard-fed ticks could feed for almost a week. And then we see higher molting rates in our lizard-fed ticks. So they're not only able to feed just a vast amount of ticks, because you get a larval burden of 100 on one single lizard, they also increase their chance of survival and molting. So I think that the lizards are very good for ticks in the West Coast system for a lot of different reasons.
Yeah, very interesting.
Yeah, and and there's also a study looking at host preference where they basically put all these different hosts in different compartments and let the ticks choose which host they wanted to go to, and pretty much all the ticks went to the lizard. So there's something that is, attracting the ticks to the lizard, but we don't really know what that is yet.
Yeah, they're certainly everywhere out here.
Yeah.
All right. Well, thank you so much. Very interesting talk. And I think this was lovely having two of you back to back. I think this has been a success already. So thank you so much for your willingness to do that. And we're excited to see about the next steps in your work and your career.